Hello, everyone. Today we are going to take a look at the second major step in the process of urine formation, and that is reabsorption. And then we're also going to look at the third step, which is secretion. Remember that urine formation involves three different major steps. The first is filtration. The blood is filtered to form a filtrate. Then useful substances like electrolytes and water and nutrients are reabsorbed from that filtrate as it passes through the nephrons. And then finally, waste products that didn't get filtered can be secreted out of the bloodstream and into the filtrate that's located in the tubules um, in order to get rid of additional wastes. So the next part of the nephron after the renal corpuscle, which consists of the glomerulus and the glomerular capsule, is the proximal convoluted tubule. And in the proximal convoluted tubule, approximately 65% of all of the glomerular filtrate is reabsorbed back into the bloodstream. So these are mostly useful substances, things like electrolytes and also nutrients like glucose, amino acids, any other thing that would be of use to the body. Additionally, there's also some secretions of substances from the peritubular capillaries into the proximal convoluted tubule. So things like hydrogen ions, toxins, and certain other wastes are actually secreted. So secretion is basically just the opposite of reabsorption. It's a movement of substances out of the blood into the tubule. Reabsorption is the movement of materials from the tubules back into the circulatory system. So the proximal convoluted tubule on the inside has a very large surface area due to numerous microvilli that provides a larger surface area for reabsorption. Transport mechanisms in the proximal convoluted tubule include active transport, especially active transport of sodium and potassium. Also co-transport of nutrients like glucose and amino acids. And there's also antiport transporters that can secrete hydrogen ions in the proximal convoluted tubule. And also just simple diffusion. Water is reabsorbed in the proximal convoluted tubule just by simple diffusion. Since there's so many active transport mechanisms occurring in the proximal convoluted tubule, the cells that make up the walls of the tubule contain numerous mitochondria to supply the ATP that's needed for active transport. So the proximal convoluted tubule alone accounts for a very large proportion of a person's resting ATP consumption. Even though these tubules are microscopic, they still use up 6% of a person's resting ATP production. So, what we're looking at here next on the screen is a cross-section through part of the wall of the proximal convoluted tubule. And so this area up above here would be the hollow portion of the tubule, which would be called the lumen of the tubule. So on the other side of the lumen, this would be the interstitial space of the kidneys where the peritubular capillaries are located. The peritubular capillaries are actually just a continuation of the efferent arteriole. And the peritubular capillaries take up substances that have been reabsorbed from the proximal convoluted tubule. So, there are two different routes by which substances can be reabsorbed across the walls of the proximal convoluted tubule. The first is called the transcellular route. Transcellular route just goes right straight through the cells of the tubule. And these cells are columnar-shaped cells. The other route is called the paracellular route. Paracellular route is the movement of substances like water and electrolytes between the cells that make up the walls of the proximal convoluted tubule. So between the cells of the proximal convoluted tubule, there are actually tight junctions. There is some possible movement, though, of water and electrolytes across those tight junctions, and that's called the paracellular route. We'll talk about this a little bit later. Now, in addition, there are several different sides to the cells that make up the walls of the proximal convoluted tubule. The first is the side of the cells that are adjacent to the lumen, which is the hollow portion of the tubule, which is in the middle of the tubule. That's called the luminal membrane. And the luminal membrane has microvilli, which are little finger-like extensions or microscopic extensions. And that greatly increases the surface area available for reabsorption. In addition, there are the sides of the cells that are either between adjacent cells, in other words, the lateral sides of the cells, and also the side opposite the luminal membrane. Those membranes are called the basolateral membranes. Okay, so it's important to remember the names of these membranes because we'll be talking about the transport mechanisms that are in these different membranes. There's different transport mechanisms located in the basolateral membrane and also the luminal membrane. So, the key to reabsorption of almost all substances in the proximal convoluted tubule is the reabsorption of sodium. Because sodium drives the cotransport or reabsorption of almost all other substances that are reabsorbed by the proximal convoluted tubule. The reason for this is the reabsorption of sodium, which is by active transport, creates an osmotic gradient that drives the reabsorption of water and also the cotransport of other substances like glucose by way of symport proteins. So let's take a look at a diagram, another diagram of one of the cells or some of the cells that make up the walls of the proximal convoluted tubule. So on this diagram, the lumen is on the left-hand side. Now, you need to remember these cells go all the way around the tubule. So depending on what part of the tubular wall we look at, it may be located on different sides. So it may shift depending on what diagram we looked at. But on this diagram over here is the lumen. This is where the filtrate is located. That's been filtered in the glomerular capsule. Over here on this side, this is the interstitial fluid, and this is one of the peritubular capillaries that surrounds the nephron. So, what drives the reabsorption of sodium across the luminal membrane, which is here, is actually the active transport of sodium across the basolateral membranes. So located along the over here on the lateral side of these cells, and also on the basal side of the cells, there's sodium, potassium, ATPase pumps, which is a form of active transport. And these sodium, potassium, ATPase pumps, which are running all the time, they actively transport 3 sodium out of the cytoplasm of the tubular cell, and at the same time they transport 2 potassium to the inside of the tubular cell. Now, what that does is that increases the concentration of sodium in this interstitial fluid. So that's going to create an osmotic gradient, which is going to drive the reabsorption of water. Okay, that's one thing. Also, by transporting sodium out of these tubular cells, that decreases the concentration of sodium in the cytoplasm of that tubular cell. So inside this cell, we have a low concentration of sodium, and it always stays low. There's always a standard gradient, which is low in sodium on the inside of the cytoplasm of those cells, because these sodium-potassium ATPase pumps are always pumping the sodium out. And so the concentration of sodium in these tubular cells is always lower than the concentration of sodium in the filtrate. So again, we have an osmotic gradient, or we have a concentration gradient for sodium, where it's higher in the filtrate than it is on the inside of the cells. Because so these sodium potassium ATPase pumps create what's called a standing gradient for sodium across the luminal membrane. Okay, now in the luminal membrane there are carrier proteins that are involved in facilitated diffusion of sodium. So remember, facilitated diffusion is a form of passive transport, but it requires a carrier protein. So that's the kind of transport we have in the luminal membrane, is facilitated diffusion. But since facilitated diffusion is a passive form of transport, you have to have a gradient. So since we do have a gradient of sodium, which is higher in the filtrate and lower in the cell, that enables sodium to move across the luminal membrane by facilitated diffusion. So once sodium gets inside the cell, then it's almost immediately pumped out across the basolateral membrane into the interstitial space, where that sodium is then taken up by the peritubular capillaries. So that's how sodium is reabsorbed. Now, once sodium is reabsorbed, this movement of sodium across the luminal membrane allows for the co-transport of other substances like glucose and amino acids. So there's also other co-transport proteins or symport proteins that co-transport glucose and amino acids along with sodium. So these other substances just kind of piggyback on the sodium gradient. That transports substances into the cell. From there, they can just diffuse across the basolateral membrane, and then they can be taken up by the peritubular capillary. So the active transport across of sodium across the basolateral membrane drives the co-transport of other useful substances like glucose and amino acids, which can be used as nutrients by our body. So you don't want to lose those useful substances. Now, one of the things you may be wondering about is if these sodium-potassium pumps transport potassium into the tubular cells, what prevents the potassium from just diffusing down its gradient across the luminal membrane into the filtrate, which would cause the loss of potassium to the urine. And potassium is a useful substance. It's needed for action potentials, for example, and also for muscle contraction. So what happens is that there's leak channels in the basolateral membranes for potassium. So when the potassium gets actively transported into the tubular cells, it just diffuses down its gradient through these leak channels into the interstitial fluid, where it is taken up by the peritubular capillaries and it's returned to the blood. So that way we don't lose too much potassium. So this model that I'm showing you right here, this is called the standing gradient model, because the activity of the sodium-potassium ATPase pumps creates a standing gradient for sodium across the luminal membrane, which allows sodium to move down its concentration gradient by facilitated diffusion across the luminal membrane. And then from there, it's pumped out of the tubular cell into the interstitial fluid, and then it's taken up by the peritubular capillaries. All right, now, this is another diagram of the cross-section through part of the wall of the peritubular capillaries. This is on the other side of the tubule, though, so it's kind of confusing because the lumen on this diagram is over here on the right. It's just because we're looking at the other side of the tubule. So this tan shaded area, that's the lumen. Over here is the interstitial space. This is one of the peritubular capillaries that surrounds the nephron. So this is a real busy diagram, but what it's showing is how other substances are reabsorbed. And basically the way other substances are reabsorbed is by way of cotransport proteins. So they're cotransport or symport proteins in the luminal membrane. So when sodium diffuses down its gradient by way of these symport proteins, other substances like glucose piggyback on the sodium gradient. So that increases the concentration gradient of things like glucose and also chloride and amino acids in the cytoplasm of the tubular cells. And then useful substances like glucose can be transported by facilitated diffusion across the basolateral membrane. And then other substances like this transport protein right here, that's an antiport protein because it's co-transporting hydrogen in the opposite direction. So it's actually causing the secretion of hydrogen. So the secretion of hydrogen ions lower the pH of body fluids. So if we have too many of them, it's a waste that needs to be eliminated from the body. So one of the ways this is done is by secretion from the tubular cells in the proximal convoluted tubule. So that's where secretion comes into play in the proximal convoluted tubule. Now, in addition, because the sodium-potassium pumps are increasing the concentration of sodium in the interstitial fluid, this creates a concentration gradient for sodium. And so that draws water by way of osmosis via the paracellular route. So it can go through these tight junctions. And so water is just moving down its concentration gradient. And that results in reabsorption of water via the paracellular paracellular route. And when that occurs, there are some electrolytes like sodium, potassium, chloride, magnesium, calcium, and even phosphate that end up getting carried by that water through that paracellular route. So this process by which electrolytes, as well as urea and uric acid, get reabsorbed by the paracellular route is called solvent drag. So in other words, the water molecules are just dragging those substances across the walls of the proximal convoluted tubule. They just kind of go along with the water for the ride. The water contains these dissolved substances, and so when the water moves down its concentration gradient, these other substances end up getting dragged along with the water molecules. And so that actually causes the reabsorption of some of the nitrogenous wastes. But not all of the urea and uric acid gets reabsorbed. So enough of it does not get reabsorbed that some of it can get eliminated via excretion of urine. So even though not all of the urea and uric acid gets excreted, enough of it does get excreted to lower the concentration of those substances in the blood. So basically, reabsorption of most all other substances in the proximal convoluted tubule occurs via cotransport, utilizing that sodium gradient that's caused by active transport of sodium. So sodium pretty much drives the reabsorption of just about everything else that's reabsorbed by the proximal convoluted tubule. So as I mentioned, urea also passes through the tubular epithelium with water by way of solvent drag, by way of the paracellular route. So as a whole, the nephron reabsorbs 40 to 60% of the filtered urea, which is surprising because that's a waste. That's something we want to get rid of. But because the nephrons reabsorb 99% of all the water that's filtered, the urine ends up with a higher relative concentration of urea. So if you take out all the useful substances and leave about half of the urea in the filtrate, then the urea is going to get concentrated. So when that concentrated filtrate leaves the kidney, it has about half as much of urea as it did when it was filtered. So not all urea is removed from the blood, but enough is removed to maintain safe levels. And actually, as we'll see later, that urea that's reabsorbed actually helps create a concentration gradient in the medulla of the kidney. That concentration gradient is one of the things that allows the nephron to produce a very concentrated urine. It's what allows the nephron loops to reabsorb more water. So the reabsorption of urea actually has a side benefit, as we'll see later. Now, another waste product that's produced from the breakdown of nucleic acids is uric acid. The proximal convoluted tubule actually reabsorbs nearly all of the filtered uric acid, which again is surprising because that's a waste product. Normally you want to excrete it, you don't want to reabsorb it. However, in the later parts of the nephron, most of that uric acid is secreted back into the filtrate. And so it's reabsorbed, but then it's secreted again, and then it passes out of the urine and is eliminated from the body. Creatine is another nitrogenous waste that's produced from the breakdown of creatine phosphate, which is found in muscles. That's not reabsorbed, so once it's filtered, it just passes out with the urine. All right, so the active transport of sodium into the interstitial fluid surrounding the proximal convoluted tubules increases the osmolarity of the interstitial fluid. That creates an osmotic gradient that draws water out of the tubules via the transcellular and the paracellular routes. So water transport through the transcellular route, which goes directly through the cells of the tubules, that occurs through water channels that are called aquaporins that just allow the diffusion of water through those cells. And that occurs because of that sodium gradient. So the net effect of all of this reabsorption of sodium and glucose and amino acids and other useful substance is that the osmolarity of the filtrate actually remains unchanged, because as more electrolytes are reabsorbed, more water is reabsorbed. So the concentration of the filtrate in the proximal convoluted tubule remains unchanged. It stays about 300 milliosmoles, which is about the normal osmolarity for the intracellular fluid. But by reabsorbing water and electrolytes and other substances, the volume of the filtrate is reduced by about 2/3. And it's important to realize that this reabsorption that occurs in the proximal convoluted tubule occurs at a constant rate, and it's not regulated. So it occurs automatically, no matter whether you're dehydrated or whether you're overhydrated. So this reabsorption of water in the proximal convoluted tubule is called obligatory water reabsorption. So that reabsorbs about 65 to 80% of all the water that's filtered. Okay, because water is a useful substance. If we lose too much water, we get dehydrated and our blood pressure falls. So water is something that in general our body tends to reabsorb. But the exact amount of water that is excreted can be controlled in more distal parts of the nephron, especially in the distal convoluted tubule, as we'll see. All right, so after leaving the tubular epithelium and entering the interstitial space, water and solutes, like electrolytes, are taken up by the peritubular capillaries. And that the uptake of water by the peritubular capillaries occurs mainly just through simple diffusion and osmosis. Because the peritubular capillaries are a continuation of the efferent arteriole, which is collecting blood that has already been filtered in the glomerulus. And so much of the water of that blood that comes out of the glomerulus has gone into the tubules of the nephron. So all the blood cells, all the large proteins are still in the blood that's in those peritubular capillaries. But because we've taken some water out of it, the osmolarity of that blood in the peritubular capillaries is higher than normal. So that allows the water that's been reabsorbed into the interstitial space to be taken up by the peritubular capillaries by osmosis. Also, electrolytes like sodium and potassium, they're taken up just by simple diffusion because, again, much of the sodium and potassium was filtered in the glomerulus. And so the concentration of sodium and potassium in the blood in the peritubular capillaries is lower than in the other blood vessels of the body. So most of the substances are just taken up by osmosis, and also some electrolytes and other solutes are taken up by solvent drag along with the movement of that water. Some other factors that facilitate the uptake by the peritubular capillaries is the accumulation of fluid on the basal side of the tubular cells, which creates a higher fluid pressure that drives water in the peritubular capillaries. Also, the efferent arteriole has a diameter that's relatively narrow, which lowers the blood hydrostatic pressure, lowering the resistance to reabsorption, and also because of the high colloid osmotic pressure of the blood in the peritubular capillaries, which facilitates the uptake of water. Colloid osmotic pressure is the osmotic pressure that's caused by suspended particles in the blood, like the blood cells and platelets, as well as large proteins. Now, one thing to keep in mind about reabsorption of useful substances like glucose and amino acids is that in order for glucose or amino acids to be reabsorbed, they have to bind to a symport carrier protein. or a co-transport protein. So if your filtered load of glucose was really high, then the filtrate's going to have a high concentration of glucose. If you get a really high concentration of glucose in the filtrate, then that's going to saturate all of those import proteins that are involved in the co-transport of glucose. And if all those transport proteins are occupied, then you're not going to be able to get any further increase in reabsorption because they can't move across the luminal membrane just by simple diffusion. You have to have a carrier protein. So there's a maximum rate of transport across the cells of the proximal convoluted tubule. That maximum rate of transport that's caused by the limited number of transport proteins is called the transport maximum. So most useful substances like glucose and amino acids have a transport maximum. So if the concentration of glucose in the filtrate exceeds the transport maximum, then you're not going to be able to reabsorb all the glucose that's filtered. And so instead, some of that glucose is going to be excreted in the urine. Now, in most healthy people, almost all the glucose and almost all the amino acids is reabsorbed by the nephrons. But if someone has diabetes mellitus, then their blood concentration of glucose is going to increase much higher than normal. And so when that gets filtered in the kidney, the filtrate is going to have a much higher concentration of glucose. If that exceeds the transport maximum, then that excess glucose is going to get excreted in the urine. And so that's why patients that have diabetes mellitus, whether it be type 1 or type 2, that's why they have what's called glucosuria, which is glucose in the urine, because the concentration of glucose in the filtrate has exceeded their transport maximum. So ordinary glucose has a transport maximum of 320 milligrams of glucose per minute. In healthy people, the filtrate enters the tubules at a rate of 125 milligrams per minute. So nearly all the glucose is reabsorbed in healthy people. But in patients with diabetes mellitus, the glucose concentration in the blood may reach 400 milligrams per deciliter, which would exceed the transport maximum for glucose of 320. So any glucose that doesn't get reabsorbed is excreted in the urine. Now, tubular secretion is the transport of waste and hydrogen ions into the tubules of the nephron. Urea, uric acid, bile acids, ammonia, catecholamines, prostaglandins, and some creatine are secreted. Also pollutants, drugs like morphine, penicillin, and other drugs are also secreted. So secretion of hydrogen ions in the proximal convoluted tubule and also the distal convoluted tubule help maintain acid-base balance of body fluids. In the proximal convoluted tubule, secretion of hydrogen is obligatory, and it is driven by the concentration gradient of sodium that was produced by those sodium-potassium ATPase pumps in the basolateral membrane. Now, we're going to talk a little bit more about secretion in the distal convoluted tubule and the collecting ducts. Collecting ducts are the last part of the nephron. There are two kinds of cells. There's principal cells that have hormonally regulated transport proteins that regulate salt and water reabsorption. That's under the control primarily of the hormone aldosterone. And then there's intercalated cells. The intercalated cells have potassium, hydrogen, antiport proteins that secrete hydrogen and reabsorb potassium by facilitated diffusion. So suffice to say, you also get more secretion of hydrogen in the distal convoluted tubule. And that takes place within the intercalated cells. So, in summary, and just to wrap up this section of chapter 23, approximately, it may vary slightly, but approximately 65% of sodium is actively reabsorbed by the basal lateral membrane of the proximal convoluted tubule. That reabsorption of sodium creates a concentration gradient, which drives the reabsorption of water by osmosis and reabsorption of other useful substances like glucose by cotransport mechanisms. Also, about 50% of the urea, which is a waste, is reabsorbed with water by way of solvent drag. That's via the paracellular route, leaving the other half to be excreted, so that 50% of the urea in the filtrate that's excreted, that's usually low enough to maintain safe levels of urea in the blood. Also, once solutes in water enter the interstitial fluid, they're then reabsorbed by the peritubular capillaries by osmosis or solvent drag. Most reabsorbed solutes have a transport maximum due to limited transport proteins, and secretion of hydrogen, as we saw, occurs in both the proximal convoluted tubule as well as the distal convoluted tubule. So, that's all I have for you today. As I said, urine formation and the physiology of the urinary system is a rather complex process. So be sure to read your textbook, or be sure to read your smart book on Connect Plus. And also don't forget to do your Connect Plus questions. I have extended the deadline for the Connect Plus questions. So you have until the 30th, which is the date of our next exam. So remember that our next exam is going to be at 1 o'clock on the 30th. So just stay tuned for an email that I'll send out where I'll let you know what you need to do for that exam. All right, I'm working on the exam now. I'm trying to get it set up on Canvas, so there'll be more details later. All right, well, I'll see you later.